So it's my pleasure to introduce Jim Hendee.
Good morning. I have a lot to cover, so I'll try to go quickly. First of all, I'd like to thank Gustavo for allowing us to use his presentation, well, not allowing it, but giving us his template for use on the presentation, and Chris Kelble, my deputy, for supplying quite a bit of what we have on there. I'm the director of the Ocean Chemistry and Ecosystem. Oh, it wasn't clicked on the.
Yeah, when you're on the GoToMeeting part.
I see. Okay.
There you go.
Okay, our vision would be to, excuse me, be a world leader in understanding the impact of anthropogenic and natural stressors on coastal and marine ecosystems, and in understanding the provisioning of services from these ecosystems. We apply this knowledge to enhance coastal and marine ecosystem sustainability and resilience. To accomplish our vision, we will quantify the role of oceans in sequestering carbon dioxide, characterize, understand, and predict the impact of pressures and their synergistic interactions on marine ecosystems, and develop products to enhance coastal and marine resiliency and sustainability.
1 second, Jim. I realized I just got a text message that we are not showing on screen. Okay. There we go, and we should be good.
Okay, hopefully everybody online can see us. Okay, our divisional objectives. We will accomplish our goals with diverse world-class workforce, which you will see. Our cutting-edge research, which I think you saw some of yesterday at our aquarium, excuse me, our Reef lab. And also you'll see some more today at the Advanced Design and Manufacturing Laboratory. Our technology development, you'll see how we have developed a new sea temperature sensor. We intend to communicate our science and our accomplishments to not only stakeholders, but to other scientists through peer-reviewed publications and to our NGO partners. Speaking of partnering, we intend to partner with NGOs, other scientists, other organizations, and other line organizations in NOAA. We are going to concentrate more on transitions. It's something that we haven't done so much in the past, but we're doing real well at that now. And we need to secure funding so that we can keep growing our laboratory. You'll see a little bit more about that coming up. I have 41 geniuses working for me. This is comprised of 14 federal employees, 25 Cooperative Institute employees, 20 scientists, 20 science support, 5 Siemens postdocs, 3 graduate students, 1 National Research Council postdoc, and I'm happy to say that we've been able to get a NRC postdoc just about every single year for the last 10 or 15 years, and 1 NOAA Corps officer, and we've always been fortunate to have 1 or 2 NOAA Corps officers working with us. Okay, I'm going to go over 5 main research themes that we do our work on. I'll start with the ocean carbon cycle. Rick Wanenkoff, who is our ST scientist, started here almost 30 years ago, and he and Richard Feely over at PMEL really started the ocean carbon cycle and have done really well at going at all the oceans around the world and studying PCO2 and ocean carbon. And in the meantime, we have acquired Dennis Perot, who will be talking next, Letitia Barbaro, and she'll be talking about her understanding ocean acidification at the regional scale in the Gulf of Mexico. We study ocean carbon inventories and fluxes, sustain global and regional carbon observations, seasonal to decadal variability and CO2 update, surface ocean carbon dioxide observing network, and the global and coastal ocean carbon modeling and synthesis. About the same time Rick started, 30 years ago, I started the Coral Health and Monitoring Program. And since then, I was fortunate to be able to hire Derek Manzello shortly after that, about 20 years ago. And he hired Ian, whom you saw, won the silver medal. And so they've hired a bunch of really great people working for them, and so we have a very strong coral program. We study coral reef bioerosion, coral calcification, corals in marginal environments, and the National Coral Reef Monitoring Program, which your colleague, Steve Thur, was in charge of the Coral Reef Conservation Program when that came about. I think we all got together in Key West and decided on the master plan, and I handed it over to Derek, and he's taken it. He and Ian have done really well with that. Molecular mechanisms and source tracking. Cristina Galliano does the microbial source tracking technology here at AOML. And decadal projections of coral bleaching. Ruben van Hoedonk, whom we hired, is one of the leading predictors of by decades of coral bleaching. There aren't too many people who do that, and he was in the recent IPCC report. Another major research area is ocean acidification, especially mesophotic CO2 dynamics or coral reefs, ocean acidification product suite that we've been doing for a while, coastal and marginal seas ocean acidification network, including the Gulf of Mexico ecosystems and carbon cruise study, ships of opportunity for ocean acidification, the Global Ocean Acidification Observing Network, the Gulf of Mexico PCO2 and Ocean Acidification Database, quantify ecosystems impact of ocean acidification, and effect of ocean acidification on calcification and erosion in coral reefs, which is what Derek studies quite a bit on. Omics. Okay, if you know Kelly Goodwin, she's been pushing omics like a D9 on the transatlantic pipeline, and she's finally got it funded. She and others, of course, funded every year now. We started this with fisheries, we AOML and fisheries, and now it's doing some great sciences we'll outline here shortly. Kelly, in fact, has done some autonomous omics platforms at GLERL, Great Lakes Environmental Research Laboratory. We do eDNA to support ecosystem understanding using indicators and modeling. Ichthyoplankton metabarcoding, in situ harmful algal bloom, toxin warning. Green turtle monitoring. Now this I really like. This is something Kristen Harper does. She's a postdoc working with Kelly, and she is working on detecting green turtles just by taking these water samples. This is a really nice thing. Pathogens potentially associated with red tide. This is something Cristina Galliano is working on. The Red Tide Group has been working with the fishermen, the Fishermen Watermen's Association. And it turns out that some people are complaining of getting diseases associated with red tide, not really red tide itself. So Chris is going out with the cruises and talking to the fishermen and trying to find out more about that. Larval genetics and proteomics of heat-resistant coral genotypes, and I believe Anderson Mayfield is working on that quite a bit. Another major research area that Chris Kelbel, the deputy here, is working on. This aims to establish socio-ecological status of coastal and marine ecosystems, community resiliency to red tide, national marine sanctuaries, making sure that they have an ecosystem-based fisheries management plan. We continue to work and have worked for many years on the South Florida Ecosystem Restoration Program. And Dr. Jiazong Zhang, there in the middle, he works on eutrophication and early warning signs, as well as Chris and his postdocs also work on that. Okay, a bit about our divisional funding. Sorry to say it has dwindled somewhat over the last 10 years, but we've been able to keep our head above water by the number of proposals we write that are funded. So actually, we've been able to write 20 proposals that got us $5.3 million to continue our omics and our research work across the street, as well as some bioinformatics work, which I'll touch on. Okay, there's a lot of text here, but these are major observational activities that we have been doing for quite a long time, and there are some new ones here too, of course. The repeat hydrography has been going on on the global ghost ship or hydrographic cruises, as we call them. The Gulf of Mexico ecosystem and ocean acidification cruises, the Ship of Opportunity PCO2 for the Surface Ocean Carbon Atlas, and the National Coral Reef Monitoring Network, which I mentioned before, has been established in Puerto Rico, the Florida Keys, and the Flower Gardens banks. It's called actually the Flower Garden Banks National Marine Sanctuary. South Florida Ecosystem Restoration Research, small and regional research, and this new program of the Red Tide Monitoring in partnership with the Florida Commercial Watermen's Association, or Conservation Association, Water Quality and Oceanography, and eDNA on fishery surveys, California Current. This is what Kelly's been working on, Kelly Goodwin, and the Gulf of Mexico, and the characterization of marginal reef environments that Ian Enox works on for high CO2 and high urbanization, high nutrients. They're not all coming up. Oh, maybe I got to push it twice. There we go. Everybody's got all these beautiful logos. Well, we want to enhance the fact that we work with the private sector, the government entities, NOAA, academia, and international. And I'm not going to read all those out. We have to leverage our resources, especially for ship time. Thank you very much, Nancy. We have to use partnerships, nonprofits, academia for cruises, equipment deployments and logistics, federal, state, academic, and industry collaborations, pool resources with partners to extend and strengthen research cruises and enhance temporal and spatial observations, for example, red tide, and cargo and cruise ships, collaborate with marine industry to provide space for our thermosalinograph and PCO2 installations. And I'm happy to say I got to go on one of those cruises, and it's really nice to interact with the people on the cruise that know nothing of oceanography and CO2 and why we're doing this. So that was quite a good experience. And like Gustavo, when he showed the interaction among the different divisions, we have interactions with HRD and PHOD. Hurricane impacts on coral reefs. Derek published a paper on this, which is really widely cited. Role of hurricanes and red tide. This is something that we intend to work on as we expand upon our red tide research. Repeat hydrography. I think these are the same as what Gustavo put up. Thermosalinograph observations in support of PCO2 assessments. Ocean conditions during red tide events, that's new. Climate integrated ecosystem assessments and biogeochemical Argo. This is something that we're just now starting. And the impact of hurricanes on oxygen and PCO2 in the ocean. I'm going to take a decompression stop here when I'm talking about OMAO for fleet and charter, and just want to point out that Nancy Hahn used to work here as Nancy Ash, and she and I did one of the very first installations of a in situ PCO2 instrument in Discovery Bay, Jamaica. That was fun. So we work with OMAO to charter for fleets and charter to do our observations at sea. With NEMS, we work on the Biscayne Bay habitat focus area. We do omics for ecosystem-based fisheries management, coral and stock assessment. And with NESDIS, we work on state of the climate, and we do the ship of opportunity, ocean acidification, and CO2 cruises. Independent ecosystem assessments in South Florida, ecosystem restoration, and NCREMP, the National Coral Reef Monitoring Program we do with NESDIS. And of course, I mentioned the big red tide work we're going to be doing. Okay, I'm not going to read all these. I just want you to see that we're emphasizing our work in ecosystems, hence the name of our division, from Ocean Chemistry Division to Ocean Chemistry and Ecosystems Division. And of course, omics is our big push. This has really afforded us a lot of good laboratory expansion and some good work and hiring some very good people. And of course, ocean carbon goes on forever. Okay, this is, we don't have as many people as the other divisions, but we've been doing pretty well in turning out peer-reviewed manuscripts, and that's because of the good people we recently hired. I think the dip there in 2013 to 21 is when Derek took a vacation. Only kidding. And so we expect to be able to keep on producing good papers. Okay, I'm going to talk about these key areas of research a little more. The coral restoration omics. The relevance are coral reefs are highly valuable ecosystems that are threatened by a number of stressors. Here in Southeast Florida, coral reefs are worth about $80 billion and 70,000 jobs. So that's just a piece of the iceberg, so to speak. And what we are doing is experimental observations, genomics, transcriptomics, and proteomics. And what we have learned is there are more resilient genotypes, transcriptomic differences, and putative pathogens between diseased and controlled acropora. And there are hundreds, or at least 100 species of acropora, but we only have 2 here in South Florida. Current work, coral health assessment or pre-death, omic investigations into resilience and marginal environments. Okay, this is a key area of research, detecting eutrophication before regime shift. I had the pleasure of being able to read this or review it before it went out. This was one of Chris Kelbo's postdocs who did this. Eutrophication is a global problem that is difficult or impossible to address after it causes this regime shift. And what we're doing is developing statistical methods to detect long-term trends in nutrients in chlorophyll A and oligotrophic systems. What we have learned is that we can detect this spatial chlorophyll A and nutrient trends that signal a likely regime shift. So the plan is to apply these results to effectively manage land-based sources of pollution in Biscayne Bay habitat focus area right here next door. Another key area of research is the changing ocean carbon cycle. We all know the planet is warming up and it's all because of carbon. Well, several other things. Okay, we want to understand and predict changes in ocean climate and impacts of ocean health. What are we doing? Obtain accurate ocean carbon data, increased understanding of physical and biogeochemical processes to constrain ocean carbon inventories, fluxes and trends in ocean acidification, and improve methodologies, autonomous platforms, and sensors. The work we do now is we are the largest data provider to the Southern Ocean Catalog. I forget. Right. I always forget that one. And lead regional ocean acidification cruises. With PMEL, we lead NOAA component of the Ghost Ship, the hydrographic survey, collectively use data, remote sensing, and artificial intelligence schemes to provide monthly near real-time air CO2 fluxes. And on the right, you can see this recent paper that used AI for a monthly annual air CO2 flux product. Okay, another key area of research is the coral monitoring research and modeling. The relevance is, as we mentioned, coral reefs are highly valuable ecosystems. They support economies throughout the world. Wherever there's a coral reef, there's usually a fishery associated with it that the indigenous people rely on. What we are doing is laboratory experiments, field observations, modeling, 3D scanning. I think Erica might be able to point out to you our 3D scanner when she's giving you a tour of the boats later on. And what have we learned? Ocean acidification affects coral and algae species richness. Ocean acidification decreases calcification and increases bioerosion. Business as usual. In other words, if we keep doing things like we're doing and we don't join the, I better not get into politics, scenario, it will result in annual severe bleaching by 2040. There is an effect of genotype and environment on restoration success. Our current work, land-based sources of pollution impacts in South Florida, nursery resilience scorecard, reef framework persistence model, coral ecosystems and marginal environments, and long-term coral and ocean acidification monitoring. Key area of research that we do, of course, is environmental DNA. And what's really nice about this is we can collect massive amounts of data just by collecting water. What we are doing is biodiversity assessments from microbes to whales, pathogen abundance, coral holobiome, and by that we mean not just the coral and the zooplankton that live inside of there, but also the microbes that live around there. The potential ecosystem indicator evaluation and use for ecosystem modeling. Oops, I think I went ahead on that one too quickly. We have learned eDNA can detect organisms that divers cannot. eDNA varies temporally and spatially along known biogeographies. eDNA can detect zooplankton, phytoplankton shifts in the California current, which is of course important, where Kelly's working with the huge anchovy harvest that we have there. And our plan is sample eDNA on ongoing research cruises to answer pressing questions, develop technology to sample eDNA autonomously on ships, and add eDNA to ongoing ocean cruises, for instance, our Ghost Ship hydrography. Okay, a transition, we have a sanctuary conditions report. Relevation, NOAA's National Marine Sanctuaries would like to use these indicators in their condition report and ensuing management plans. The objective is to develop a method to select indicators for condition reports and pilot the implementation of indicators into the Florida Keys National Marine Sanctuary management process. What we have learned is indicators are developed and analyzed for all condition report questions. What is transitioned is indicators were used in the draft environmental impact statement by the Florida Keys National Marine Sanctuary. An indicator selection process is currently being transferred for use in the Olympic Coast National Marine Sanctuary. This is something that Kelly Goodwin has worked on, this transition of EAUVs, develop autonomous systems and further eDNA approaches to advance ocean sciences and stewardship and support the blue economy. Objective is to demonstrate proof of concept to show the feasibility of a prototype instrument, EAUV, to collect and process samples for molecular biological analyses. What we have learned is utilize deployment missions to define operational requirements and inform engineering developments to increase the readiness level of the instrument, which we have done in moving the EAUV from RL4 to RL5. Another transition, molecular microbial source tracking. Some of the U.S. territories, such as the Commonwealth of Northern Marianas Islands, need microbial source tracking capacity to better manage and mitigate land-based sources of pollution impacts in coastal waters, which is of course important because small islands get lots of people and therefore lots of pollution, and it's got nowhere else to go. Adapt qPCR instrumentation for microbial source tracking analyses, train personnel with their own in-house instrumentation. And what we have learned is microbial source tracking capability is used to conduct a pollution source tracking study in Saipan Lagoon, and that's a great place to dive, by the way. Adaptation and modification of qPCR as part of our transition, training of lab personnel and microbial source tracking methods, ongoing consultations between AOML and CNMI for land-based sources of pollution studies. I'm going to skip over this because you're going to be seeing this next, the Advanced Manufacturing Lab, except I want to say that we are going to be developing our bioinformatics computing and omics laboratory physical structures. You've already seen the experimental reef laboratory across the street. You know why we need it, but this was made not just for corals, but also for smaller organisms. A project that I've been involved in is development of this low-cost sensor that only costs $9 as opposed to a commercial $130 sensor. And the reason we did this is we want to deploy these at 200 coral reefs at 3 different depths around the world. And this will inform satellite monitoring, which can only measure the surface of the ocean. And we field tested this off of Southeast Florida 2 years ago and just published it in Bulletin of Marine Science. And Ian Enox and Nathan Formel of CMIS just won the DOC silver medal, which I think we announced our award ceremony yesterday. It's a low-cost open source autonomous subsurface sampler. We need it because of dial variability is often not adequately sampled and can be quite large. Without an understanding of dial variability, data cannot be put in proper context. Increased applications, modified to enable Sturbeck filter collection for eDNA sampling and so on. In fact, another nice thing that Ian has done with this is put this online for the kiddies. So it's an introduction to chemistry and ocean acidification, science and technology, and how to build a small one. And we have some great division-sponsored Rasmus graduate students, some divisional interns, and our challenges ahead are base funding. As I mentioned, it's been pretty much level for the last 10 years. Infrastructure, we're about to build on our omics and our bioinformatics computing. Our FTEs are dwindling, but we're hoping that if we do get funding, we'll be able to increase on that. We're also hoping for some retirement soon. Like everybody, we have the problem of talent recruitment and retention. But everybody here has a good time, and we have some good resources for doing good work. So as long as you like what you like to do, this is a good place to work. And ship time, as I mentioned, that's really hard because you have to program years ahead, and the ship may be on the other side of the planet and all those kinds of challenges. And transitions is something that we're trying to do more of as an operational partner. Quickly, these are the last 2 slides. We are engaged in the Argo program, but with BTC Argo, we plan to add 6 ocean core ocean variables, chlorophyll, fluorescence, particle backscatter, oxygen, nitrate, pH, and radiance. Regional pilot site where we're going to do this is the Gulf of Mexico. The advantages are OSAT experience with the BTC Argo parameters, and we need to increase AOML capacity with BTC observations, models, and data assimilation. Finally, focus on our future. Critical presence in marine chemistry and ecosystem science we have within NOAA and throughout the scientific community. We have a new generation of early career scientists that are very bright, engineers and support personnel, emphasis on social sciences. This is something we're especially engaged in with the independent ecosystem assessments, cutting edge NOAA-oriented research and transitions applications for societal benefits in the blue economy, state-of-the-art capabilities, and artificial intelligence and machine learning. This is, I set up recently a artificial intelligence listserv, in fact, and that's starting to come back around. And I think I wrote the very first artificial intelligence application back in 1993, except for National Weather Service. And that's it. We are the Ocean Chemistry and Ecosystems Division.